Welcome back everyone to another MCAT ACE lecture. This video is going to cover the functions of the plasma membrane as well as its composition, which is made of the lipid as well as protein components. And then we will describe the fluid mosaic model, go over different modes of transport across a cell membrane, discuss the functions of proteins within a cell membrane, which includes acting as channels, enzymes, or receptors. We will go over the difference between exocytosis and endocytosis, and then finally we will discuss how the cell membrane of the adjacent cells are being connected to each other. Alright, so starting with the general functions of the cell membrane. So the first obvious function of the cell membrane is to define borders of cells. And then of course, because now there is a membrane around the cell, it will help maintain the cell integrity. It will provide selective permeability for the passage of molecules inside and outside the cell. In addition, plasma membrane has a flexible structure that allows cell to pass through narrow spaces, so flexible membrane. And a good example is like, for instance, red blood cells that can go through narrow spaces of the blood vessels. And then finally, there are receptors as well as markers on the cell membrane that helps recognition by other cells, like for instance by immune cells. And for instance, in the immunology section we just discussed how the MHC1, which is major histocompatibility complex 1 glycoproteins, are on the cell surface in order to recognize self-cells and not to attack them by our immune cells. Next, we are discussing the composition of the membranes. So basically, membranes are made of three main structures, which includes lipids, carbohydrates, as well as proteins. So we would like to first start with describing the composition, the lipid components of the cell membrane, which includes phospholipids, and each molecule of phospholipid is made of one glycerol plus two fatty acids and a phosphate polar head. And so here I'm showing you an image of a phospholipid. So here we have all the three components that I just described for you. We have a glycerol attached to two molecules of fatty acids, and then there is a phosphate polar head. And so since the phosphate has a negative charge, therefore it's polar and it would be hydrophilic, versus the fatty acids, which are chains of carbohydrates. This one is nonpolar and would be hydrophobic. Now, because of the structure of phospholipids that contains both a polar head and a nonpolar tail, it's referred to as amphiphilic or dual loving, because this molecule contains both a charged and a non-charged area. Now, because of this unique structure of phospholipids, they would try to avoid water content of the cell as much as they can. And so therefore the cell membrane will have a lipid bilayer where the polar head of phospholipids, which would correspond to the phosphate head, would be facing outside as well as inside of the cell. So here we have the extracellular space that has so much water, as well as intracellular space, which again has water. So therefore the polar heads, which are hydrophilic, are now in contact with water, while the non-polar tails, which were fatty acid and are hydrophobic, are avoiding the water content of the cell. Now, another way that phospholipids can present inside an aqueous solution is that they could form a micelle, where they would form a circular form and having the tails facing each other, and the polar head will face the external water environment. And then finally, liposome is another way that phospholipids can arrange together. And with the liposome, they would be having again a bilayer that goes circular. Alright, and so this way we have all the non-polar tail at the center, while the polar head is facing the outside environment, which has a lot of water, and another polar head is on the inner surface of the liposome, which will face the water that is being trapped inside the liposome. Alright, so that concludes discussion of phospholipids. Let's move on to the next lipid molecule that we commonly encounter in a cell membrane, and this includes the cholesterol, which is made of 4 fused carbon rings, and its function is actually to reduce permeability of the cell membrane while maintaining fluidity of the cell membrane at low temperatures. And here I'm going to show you an example of a cholesterol molecule. So there we go. You can see that we have 4 carbon rings, and again its function is to 1, decrease permeability of the cell membrane, and 2nd, maintain the fluidity of cell membrane at low temperatures. The next component of plasma membrane are the proteins. And there are 2 types of proteins that you find inside the plasma membrane. One is peripheral proteins, which can occur on either side of the cell membrane, and they usually act as either enzymes, cell receptors, or cell markers. So cell receptors, markers, enzymes, or it can also function as a cytoskeletal attachment site. And so here, if we have the cell membrane, we have the peripheral proteins either on the inside or on the outside. And then we had the cholesterol at the core of the cell membrane. And then the second type of proteins are integral proteins, which span either part or all of the membrane. So it spans the membrane. And so let me show you the example right here. So here we would have an example of an integral protein that has part of it exposed on the hydrophilic environment, and the other part is exposing the hydrophobic environment. Therefore, these proteins are also having amphiphilic properties because they have a part that is being polar and then the other part that is non-polar. Okay, so these integral proteins are amphiphilic because a portion of these proteins are lying either outside or inside the cell and is hydrophilic, and then the other portion is within the cell membrane, which is hydrophobic. Alright, now we have the final component, which are carbohydrates. So then we have carbohydrates as the third component of the plasma membrane, and these carbohydrates are always found on the external surface of the cell membrane. So they are always on the external surface of cell membrane, and their main function is for cell recognition. So here we have we can have on the external surface, we can have a carbohydrate that is attached to the phospholipid component. So this is called a glycolipid. Or it can be attached to a protein, and then in that case it would be called a glycoprotein. And these carbohydrates are functioning as cell recognition sites in order for our immune cells to recognize our own cells from the external pathogens. Okay, so therefore these carbohydrates are required for cell recognition in order to differentiate body's own cells from any other foreign cells or pathogens. And then the other function of these carbohydrates is that they are hydrophilic, and so therefore they can attract large amount of waters to the cell surface. So attract water to cell surface. All right. Now that we've described different components of a cell membrane, we can go over the fluid mosaic model. So basically this model was proposed in 1972, and this model states that the cell membrane is organized into a lipid bilayer and proteins that are able to slide past each other, which gives cell membrane a fluid characteristic, and so that's why it's called fluid mosaic model. And then since the plasma membrane are made of different components, like for instance, as we just discussed, phospholipids, proteins, and carbohydrates, therefore, this model describes the plasma membrane as a mosaic structure. So here we have a mosaic model, fluid mosaic model. And here I have provided you with an image that shows all the components of the plasma membrane. So you can see that we have a lipid bilayer. The polar head is being exposed to the internal and external environment, which usually has high water content, while the non-polar tails, which are hydrophobic, are facing each other to avoid any water. Then we have the integral proteins, which are running the entire span, or it could run part of the span of the cell membrane, and they are shown in light blue color right here. So remember, these proteins are also amphiphilic because part of them is hydrophobic, while the other part that is exposed either on the outside of the cell or on the inside of the cell, these are hydrophilic. On the other hand, we have peripheral proteins, which can be located either on the outside or inside of the cell. Then we have cholesterol, which is inside the core of the cell membrane. So here you can see that there is cholesterol molecules inside the core of the cell membrane. And then finally, we have carbohydrates that could be attached either to the plasma membrane, which is called glycolipid, or it could be attached to a protein, which is called a glycoprotein. The next concept that we would like to discuss is different mode of solute transport across the cell membrane. And so basically, if we have a semi-permeable membrane, what happens is that solutes will move down their concentration gradient until they have the same concentration on both sides of that semi-permeable membrane. So let's say that we have a high concentration of Na plus on one side and low concentration of Na plus on the other side. There would be diffusion, passive diffusion, not requiring any energy, passive diffusion of sodium into the semi-permeable membrane until we have an equal number of sodium on both sides. And so this phenomenon that I just described for you is called simple diffusion, where solute particles are passively moving through a plasma membrane that I've just shown here. So it would be passive movement of solute particles through a plasma membrane. And so considering that inside the cell membrane, so here we have, let's say that we have a lipid bilayer, and so therefore the inside environment of this lipid bilayer is hydrophobic. And so therefore simple diffusion will allow only diffusion of small hydrophobic or non-polar molecules. But then there would be limited transport of ionic and polar molecules, because these molecules don't want to go through the non-polar environment of the lipid bilayer. And so therefore the next mode of transport is called facilitated diffusion. And this is still a passive diffusion, but allows movement of molecules down their gradient with the help of a membrane protein. And so therefore this protein will shield ionic and polar molecules to allow their transport across the hydrophobic area of the cell membrane. So it will shield ionic and polar molecules from hydrophobic tails. Now the type of proteins that we can have as transporters, it could be two different types. The first type is a membrane channel. So let me go different letter. The first type is a membrane channel, which is an integral protein with a hydrophilic channel through their core that provides a hydrated opening through the lipid bilayer. So if you think of it, here we can have a channel of protein that has a hydrophilic environment inside its boundaries. So here we have now a hydrophilic environment, as a consequence of which large polar or ionic molecules can go through it from area of high concentration to low concentration, because we are still talking about passive diffusion. And one good example of these membrane channels include the sodium and potassium channels, for instance, that are required for depolarization and repolarization phases of the action potential. So you know how, if this is the lipid bilayer, then we have the tails, and so assume that here is inside the cell and the other side is outside the cell. The cells have a carrier protein, which we will discuss next, that transports 3 sodium outside the cell in exchange for 2 potassium. And this type of transport uses ATP because it transport sodium and potassium against their concentration gradient, and we will discuss that next. That active transport uses ATP in order to transport molecules against their concentration gradient. But anyway, back to the topic here. So therefore there would be high concentration of potassium inside the cell and high concentration of sodium outside the cell. Now what happens when there is an action potential? That's the time that sodium channels will open, so they are naturally closed. But once there is an action potential, then the sodium channels will open and allow passage of sodium molecules through the plasma membrane, as a consequence of which there would be depolarization phase. So the polarity, so the membrane potential of the cell will increase, and this is called depolarization phase. So let's say here we had minus 60 millivolt and here is about 0 millivolt. And then in the next step, once there is depolarization complete, the cell will have to regain back its resting membrane potential. So at this point, the potassium channels, which are closed, will open, as a consequence of which now potassium is able to leak out of cell and decrease the membrane potential. until the cell reaches its resting membrane potential, and this is called a repolarization phase of an action potential. So in any case, here what I've just shown you was an example of a membrane channel that acts as an integral protein. All right. And then we have the second type of membrane protein that can act as a carrier, whereby changing its conformation it will allow passage of molecules across the plasma membrane. And for instance, GLUT, which stands for glucose transporter protein, is a carrier protein that will allow transport of glucose and other hexose sugar through the plasma membrane, because these molecules are too big to diffuse through the plasma membrane by themselves, so therefore they need the help of the carrier protein. The next mode of transport is active transport, which we just briefly described. So this type of transport uses ATP in order to move molecules or ions or particles against their concentration gradient. Like for instance, we just described how if we have the plasma membrane here, the sodium potassium pump will pump potassium inside the cell in exchange for sodium 3Na plus pumped out in exchange for 2K plus that is pumped in, and there would be accumulation of K plus inside versus there is accumulation of Na plus outside the cell. So this is an example of where molecules are being moved against their concentration gradient with the help of cell energy, which in this example ATP has been used. Another example that I can give you is, for instance, the electron transport chain. So you know how in the electron transport chain, so let's say here we have the mitochondria, that's the outer membrane, so here it is a lipid bilayer, and then we have an inner membrane of a mitochondria. So this is the matrix of a mitochondria. We have the inner cell membrane, we have the intermembrane space, and then we have the outer membrane. Now we discussed in separate video extensively the mechanism of electron transport chain. So here I would like to briefly touch on it. So basically we have the electron transport chain components in the inner membrane. And what these proteins do is that they would transfer the electron from either NADH or FADH2. And the energy from these molecules is used in order to pump H plus against its concentration gradient. So the H plus molecules will now go to the intermembrane space and build up there. All right, and so again, that's another example of active transport because energy is used to move molecules against their concentration gradient. And then later on, once this concentration gradient built up, then these H plus can go through the ATP synthase in order to generate ATP from ADP molecule. Okay, and here I've actually provided you a drawing of the first example that I gave you. 3 Na plus molecules are being pumped out of the cell, while 2 K plus are being pumped inside the cell, as a consequence of which there is now accumulation of K plus inside the cell versus high concentration of Na plus outside the cell. And for this process, energy from ATP is used to pump ions or molecules against their concentration gradient. The last topic that here we are discussing regarding transport across the cell membrane is called osmosis, which refers to movement of water through a semi-permeable membrane. And this process is also passive diffusion. It doesn't require any energy. And so since it's passive, water must be moving from solutions with the low concentration of solutes into solutions with high concentration of solutes. And here, let me show you an example. So let's say that we have a cell that has high concentration of solutes inside. And for argument's sake, let's say that this membrane is impermeable to these solutes. So these solutes cannot move outside the cell. So what happens then in terms of osmosis? And the answer is that the water molecules will try to go inside the cell as much as they can until the concentration of both sides is the same. And so therefore, water molecules will go inside the cell and cause swelling of the cell. In this example, isotonic cell, the concentration, the solute concentration inside the cell is the same as outside. So the rate of water going inside would be equal to the rate of water going outside. And then finally, here we have an example of a hypotonic cell where the solute concentration is much lower than the outside environment. As a consequence of which, under these circumstances, water will exit the cell. And that's why if we place bacteria inside a solution or inside something that is extremely high concentration of solutes, that's why the cell will die, because all the water will get absorbed out of the cell. And then likewise, if we have a very low concentration of solutes in the extracellular environment, if too much water continue to enter the cell, it could cause the cell to burst. The next concept is endocytosis versus exocytosis. And so here first I would like to focus on endocytosis, which refers to folding of the plasma membrane followed by formation of intracellular membrane-bound vesicles that will enclose the extracellular fluid or molecule. So if we had a cell here, there would be folding of a cell membrane in order to get whatever is around here and then form a vesicle around it that goes inside of the cell. And so next I would like to discuss different forms of endocytosis that we have. Number one is fluid endocytosis, where the endocytotic vesicles basically only enclose the extracellular fluid. The second type is called absorptive endocytosis, where the extracellular molecules that are bound to the cell surface will get carried inside the cell. And both the fluid endocytosis and absorptive endocytosis are sometimes referred to as pinocytosis because they would bring inside very small molecules. Versus phagocytosis, which is also known as cell eating, is the process where folding of the cell membrane brings little extracellular fluid but more of larger molecules. And this type of endocytosis, for instance, is used by macrophages to engulf pathogens. And here I have provided you with an image that shows you all three forms of endocytosis. So there we go. The first one was fluid endocytosis, where there would be invagination of the plasma membrane in order to bring extracellular fluid inside the environment of the cell. And then we had absorptive endocytosis, where the molecules that bind to the cell surface receptors, those are being endocytosed. And then finally we had phagocytosis, which was for larger molecules. Little extracellular fluid enters, but then this type of endocytosis brings in larger molecules into the cell. All right, next we have the exocytosis. So we've already described endocytosis. The next concept was exocytosis, which refers to fusion of cytoplasmic vesicles with the plasma membrane in order to release their contents. And so here again I have another image for you which shows you how Golgi apparatus are sending vesicles to the external plasma membrane in order to release the contents. And another example that I can give you is, for instance, the nerve cells. So you know how the nerve terminal is having all these vesicles that contain neurotransmitters. So once there is an action potential, there would be calcium that influxes into the axon terminal, as a consequence of which, now that calcium concentration increases, these vesicles can fuse with the plasma membrane. So once they fuse with the plasma membrane, the neurotransmitters will be released into the synaptic cleft. And so therefore they can now bind to the postsynaptic cell. And so basically the purpose of exocytosis is, one, it allows secretion of impermeable products, like for instance hormones can be secreted outside the cell, and then it will also help replace parts of the cell membrane, because with endocytosis the cell membrane keeps getting smaller and smaller, so we need to somehow replace that membrane that has been taken off, and that is being done by exocytosis, replacing portions of cell membrane. The next concept I would like to again reiterate how proteins can have different functions. So we've already discussed how proteins can act as channels, they can act as carrier proteins, so they can act as carriers, but then they could also act as cell receptors or cell recognition site, in addition to acting as an enzyme. And basically how receptor proteins work is that they're shaped in a way that only allows specific molecules to bind to them, and consequently upon binding of these molecules to protein receptor, the protein molecule will change its conformation and induce different effects on a cell. And so let me give you an example. Let's say that we have a protein receptor for the insulin molecule. So once the insulin hormone binds to this protein receptor, the protein receptor will activate G protein and other molecules that will initiate certain changes inside the cell in order to initiate glucose absorption from outside environment. And then the other function of cell receptors is that they could act for recognition by immune cells, and we've already discussed this, how we have MHC1 molecules on the surface of our cells so that the immune cells will not attack our own cells. And then finally, proteins can also act as enzymes, an example of which include adenylate cyclase, which is involved in ATP metabolism. So once again, in addition to working as channels or carriers, proteins can act as receptors, like for instance receptors for hormones. It can be act as recognition site by our immune cells, and they could also act as enzymes. The final topic I would like to discuss here is to explain how different cells are being held together and communicate by each other. So here I have shown you different structures. The first structure is called tight junctions, where areas of plasma membrane are being stitched tightly together to prevent any movement of molecules in between the two cells. So this is an example of tight junction, where plasma membrane is held tightly together and its function was to prevent passage of molecules and ions. The next structure that I haven't shown here is called adherens junctions. The other name for which is zonula adherens, and these are basically connection of actin filaments between the cytoskeletons of two adjacent cells. So this is connection of actin filaments between cytoskeleton of two adjacent cells. Next we have desmosomes, which are holding the intermediate filaments of two adjacent cells to each other. So this is intermediate filaments. So adherens junctions were a belt of actin that were connecting the cytoskeleton of two adjacent cells, while desmosomes is connecting the intermediate filaments of two adjacent cells. Then we have hemidesmosomes, which are similar to desmosomes in that they connect to the intermediate filaments, but the difference is that they connect the intermediate filaments of cells to their basement membrane. So here you can see that the cell intermediate filament is connected to the basement membrane. So that's the difference between hemidesmosome and desmosome. And finally, gap junctions are clusters of intercellular channels that form tunnels for aqueous connectivity between two cells. So these gap junctions allow movement of ions as well as small molecules between cells. And one example that I can give you is that, for instance, smooth muscles and cardiac muscles have a lot of gap junctions. So therefore, once an impulse is being transmitted from one cardiac cell, it rapidly spreads to the rest of the cardiac cells with the help of gap junctions. Next, I would like to answer some short questions before we move on to our practice question for this lecture. So the first question is, what is the function of waxes in the cell membrane? And the reason I've included this question is that we haven't discussed waxes. So basically this is an extremely hydrophobic lipid molecule, the job of which is to add stability and rigidity to the nonpolar tail of the cell membranes. And waxes are usually not found in animals. And the second question is, what is secondary active transport? So basically, this type of transport uses movement of an ion, like for instance sodium or hydrogen, down its concentration gradient in order to move another molecule or ion against its concentration gradient. So it moves an ion down its concentration gradient in exchange for movement of another molecule or ion against its concentration gradient. So again, since a molecule is being moved against its concentration gradient, it's being considered as an active transport, except that instead of using ATP, like for instance the sodium potassium ATPase pump, or instead of using the energy that is stored in FADH2 and NADH, it just uses movement of one molecule down its concentration gradient, and from that energy it moves another molecule against its concentration gradient. Okay, so now let's move on to the practice questions. Which of the following properties allows the cold-blooded species to maintain their cell membrane fluidity in cold waters? Next question is, which of the following molecules can be found both inside and outside of the cell? The third question is, the fluid mosaic model describes the... Alright, so if you need more time, you can go ahead and pause the video. Otherwise, I'm going to start with answering the first question. Which of the following properties allows the cold-blooded species to maintain their cell membrane fluidity? So basically we've discussed how cell membrane fluidity is being maintained by cholesterol, but I don't see that answer choice right here. Decreased number of cholesterol, that would be wrong. The answer should be increased number of cholesterols. So now let's read the answer choices. Increased proportion of glycoproteins to glycolipids. No, it shouldn't have any relation in terms of proportion of glycoproteins to glycolipids. Increased proportion of peripheral to integral proteins. Again, that has nothing to do with the fluidity of the cell membrane. And so we are left with the last choice, increased proportion of unsaturated to saturated fatty acids. And so what happens with the unsaturated fatty acids is that they have a double bond, as a consequence of which their structure won't be linear. So you know how with the saturated fat we can have linear molecules, versus with unsaturated fat, since there is a double bond, there would be a curved shape to it. And so if you think of it, it would be easy to stack these straight molecules on top of each other. And so that's why with a saturated fat we always get solid fat at room temperature, while when we are working with unsaturated fat, these are bulky and it's hard to put them on top of each other, as a consequence of which at room temperature they would have a liquid or oil form. And so for this question, if we increase the proportion of unsaturated fatty acids, again, it would add more fluidity to the plasma membrane. And so in fact, that's one of the mechanisms that cold-blooded species maintain their cell membrane fluidity, is due to the increased proportion of unsaturated fat compared to the saturated fat. If this answer choice was saying increased number of cholesterol molecules, that would also have been a correct answer. The next question is, which of the following molecules can be found both inside and outside of the cell? So we just discussed that glycoproteins are only found on the external environment, so that's incorrect. Integral protein, we described how it can span the entire length of the cell membrane, and it could be found either on the outside or inside. So here in this image, you see that the integral membrane can be both on the inside and outside, and that the peripheral proteins can also be on the inside or outside. But glycoproteins are only found outside the cell. So therefore, the answer here would be choices 2 and 3 only. The next question is, the fluid mosaic model describes the... So before looking at the answer, we described that this model explained that the fluidity of the cell membrane is because of the phospholipid and protein sliding past each other. And then the mosaic component of it is because a cell membrane is made of different components. Like for instance, it's made of a phospholipid, protein, carbohydrate, cholesterol, and so on. So now with this information in mind, let's try to answer this question. This model describes the fluidity of proteins in a lipid bilayer. That's correct, because here we are talking about how protein and lipids can easily pass each other, giving the membrane a fluid texture. Next, pattern of carbohydrates on proteins. Note that was not what was discussed by the fluid mosaic model. Fluidity of phospholipids in a protein monolayer. No, we didn't talk about protein monolayer. It doesn't even make sense what it's talking about here. And then lastly, amphipathic nature of integral proteins. That also doesn't explain fluid mosaic model. Fluid mosaic model is only focusing on the composition, which is a variety of components that gives it its mosaic structure, and that there is protein and phospholipids that can easily slide past each other. So therefore choice A would be the correct answer. All right, and then let's move on to the last set of questions. Which of the following processes does not require energy? And then the final question is, presence of which of the following cell junctions permits homogeneous contraction of heart muscles following depolarization? So if you need to pause the video, go ahead. Otherwise, let's go through the answer choices here. So which of the following processes does not require energy? We just discussed how with the simple diffusion, molecules are moving from area of high concentration to area of low concentration by directly going through a plasma membrane. So this process does not require energy. Active transport, on the other hand, it does require energy in order to move molecules against their concentration gradient. So choice 2 would be incorrect. Next we have facilitated diffusion, which also moves molecules from area of high concentration to area of low concentration, except that here it gets help from either protein channels or carrier proteins. And then finally we have osmosis, which is very similar to simple diffusion, except that instead of having different molecules that are moving from area of high concentration to area of low concentration, with osmosis we are talking about water molecules that are moving through a plasma membrane. And this process is also passive and does not require energy, because water will move from area of low solute concentration to area of high solute concentration. Like for instance, if you have a plasma membrane here, and we have water that has a low solute concentration of, let's say, 1 millimolar salt versus an area of high solute concentration of 10 millimolar salt, water will move from this area of low solute concentration to area of high solute concentration in order to equilibrate molarity on both sides of this membrane. And finally, the last question, presence of which of the following cell junctions permits the homogeneous contraction of heart muscles following depolarization? So we've already just discussed how gap junctions allows movement of ions from one cell to another. So once we are having an action potential can quickly spread to adjacent muscle cells with the help of gap junctions. And then another example is smooth muscles, which also have a lot of gap junctions that allows uniform and coordinated contraction. In contrast to heart and smooth muscles that have gap junctions, skeletal muscles do not have gap junctions. Alright, and that concludes the discussion of this video. We have covered the topics for the plasma membrane. Hope you found this video helpful, and we will see you for the next lecture.